Salam and a very good day to everyone. My name is Iza Nazira from Group 4, and today my teammates Andrew, Mardia, Muhammad Haris, and Muhammad Kodila will be delivering our presentation on exploring potential soil bacteria for sustainable wheat production. So, what was the purpose of the study? Wheat is a staple crop in Pakistan. However, the poor soil, lack of irrigation, and inefficient fertilizer use in the region prevents growers from reaching the maximum potential yield of this crop. So, the purpose of this study is to study the effectiveness of plant growth promoting bacteria, PGPR for short, in maintaining wheat productivity, is assessed as a greener alternative for synthetic fertilizer. So, what were the specific questions addressed and how were they addressed? So, a specific question addressed in this study is the specific measures taken to maximize the yield of wheat in the country of Pakistan, as the soil characteristics of Pakistan does not actually promote the productivity of crop growth. So, in this study, plant growth promoting assay was assessed, and biochemical characterization of soil bacteria PGPR was studied to see if they were a suitable replacement for synthetic fertilizers. And how were these questions addressed? So, the questions were addressed by assessing the inoculation results of native rhizospheric bacteria on wheat, on the crop growth and productivity under both the in vitro and in vivo conditions.
Hello, I am Haris. I will proceed the presentation by explaining the methods that are being used in this study. So the first method is by isolation and screening of soil bacteria. This study used dilution plates to isolate bacteria from wheat sandy loam rhizospheric soil, then grew them for 48 hours at 28 degrees Celsius temperature. And afterwards, single bacteria colonies were selected and stripped onto the TSA medium plates. Second method is by promoting assay and biochemical characterization of soil bacteria. These standard procedures were used to determine strains, ACC diaminase activity, cytophore, and biolog by using orthophosphoric acid region with spectrophotometer and then obtain the protein calibration curve. And the next method will be the identification of bacterial strains. The strains were identified by 16S RNA gene sequencing, and the sequencing was used to identify strains, followed by fatty acids, metabolite usage profiling, and nomenclature determination. The fourth method is an experimental method under control and field conditions. A 1-month growth chamber experiment in trays with sterilized soil was conducted to test all isolate strains in a growth chamber. A month after germination in a growth chamber and a month later in the pot and field trials, shoot lengths, root lengths, and weight were measured. The fifth method is called whole cell fatty acid analysis. All 10 strains were grown on TSA plates for 2 days at 30 degrees Celsius temperature. The MIDI-Sherlock software was used to determine cellular fatty acid compositions. Strains were harvested and fatty acid methyls and esters were made. And finally, statistic was used to compare the means of the crop data obtained with a significance level of 0.05 for the LSD test.
Hello everyone, I'm the presenter Muhammad Kalafidaus. So I'm going to talk about the result that we obtained for the research paper. So actually, there are 4 PGP traits that were observed in this experiment actually in the test string, which is the production of the indole triacetic acid IAA, the solubilization of the insoluble tricalcium phosphates, and the EAC activity, and also the cytophone production. So actually, all strains that were produced are with tryptophan and also without the addition of tryptophans. But for the solubility, the maximum drop in pH was observed in case of the strain 7 RE7 up to 4.38 from the initial pH of the 7 during our 7 days of incubation for solubilized insoluble manure phosphates. So actually, for the EAC dominance activity, for plants, actually to have a lower level of ethylene exhibition was important. Thus, plant PGAC dominance activity actually was considered as a efficient marker for its reduce the level of the ethylene. And then out of the 10 strains that we obtained actually, 5 of them that we have identified using the CTS RNA identification from the genus Pseudomonas, which is the RA first, 4, 6, 8, and 10. And also that Actually, four of them also come from genus Bacillus, which is RH3, 5, 7, and 9. But only one strain that belongs to the genus Alkali genus, which is the RH2. So next, I'm going to talk about this is the table 1, which is the pharmacophore traits of strain isolated from the rhizosphere. So actually, for the table 1, you can see here I am talking about the RH7 for the insolubility. You can see here the pH is the is the 4.38, and also this is the ASC dominance activity and the cytophrone activity, which is for the cytophrone activity they have been considered as the plus. 3 plus me is very good, for the 1 plus is just not good. So actually you can see a lot that have the 3 plus. But then for the next result, which is for the table 4, for the point number 4 of the method, we cannot obtain for that because it has minor error for the explanation, and also the table that obtained was not really accurate. So I think this is the best result that I could extract from the research paper. That's all from me. Thank you.
Hi everyone, I'm Madra and I will continue the presentation on what was particularly striking about the study and its finding. After reading the journal, what we can say is that the topic is interesting. Bacteria's potential for enhancing wheat output was particularly striking. When we mention bacteria, the first thing that comes to mind is that they are filthy and potentially harmful. Indeed, bacteria have a lot of beneficial properties that benefit humans and also plants on the planet. We need to switch our thinking about bacteria from ew to wow because they are awesome. Every food web and every food chain on Earth begins with bacteria. They have crucial roles in producing food, decomposing waste, and performing many ecosystem services, including enhancing the growth of plants. PGPR benefits on growth and the productivity are well known and have been correlated to phytohormone synthesis and increased nutrient supply. Next, there are also some findings about the new type of bacteria that we are not familiar with. Several genera, such as Azospirillum, Enterobacter, Buchalderia, and many more, are promising biofertilizer choices. When new types of bacteria are found in soil, new knowledge can be applied, including the invention of new technology which can improve the application of the bacteria in soil. Plant growth promoting rhizobacteria (PGPR) are a class of free-living bacteria that have been shown to increase plant growth and agricultural output via a variety of processes. GPR can create hormones that promote plants' development, increase the availability of nutrients, atmospheric nitrogen, act as biocontrol agents, and improve soil structure. Through the use of plant growth promoting rhizobacteria (PGPR), growers may be able to reduce their reliance on synthetic fertilizers and boost crop sustainability. Decreasing the amounts of chemical fertilizer and pesticides are good for the environment. Excessive usage of inorganic fertilizers has serious ecological consequences, and hence biological alternatives such as PGPR may be encouraged to avert further environmental damage. This study demonstrates that PGPR can sustain wheat production despite requiring less fertilizers. It has been found that when 75% fertilizer and 2 PGPR inoculants were used in greenhouse environments, the dried weight of tomato was much greater than when the full recommended dose of fertilizer was used without PGPR inoculants. PGPR have revealed that it can promote the plant growth by producing ACC deaminase as it decreases the levels of ethylene in developing plants' root by producing plant growth hormones. It inhibits antagonistic activity against phytopathogenic or soil-borne pathogens by producing cytophore and promotes mineral phosphate solubilization along with other nutrients.
Hi, my name is Andrew and I will be presenting about the questions did this study not answer, and I will be giving some of the suggestions for the questions. Okay, so for the first question, is this method specified for only crops like wheat only? Is it capable for other crops like potatoes, tomatoes, sweet corn, and paddy? So from the articles, the experiment is conducted for the wheat species. from Pakistan, and the experiment results shows the positive impacts on the weight, including crop growth and yield. At the same time, it reduced the weight of inorganic fertilizer with the presence of PGPR in the soil condition. As for recommendation, it is recommend to using the best field condition with different types of crops such as potato, tomato, sweet corns, paddy, and etc. Okay, so for the second question can be found on this study is, do we need to consider the soil condition, weather conditions for the PGPR to grow? So is this method suitable for the countries that have 4 seasons? As mentioned in the journal, the experiment was conducted in Pakistan as the soil was so poor and due to low organic metals content, which corresponds to the low soil fertility and poor soil structures. So as recommendation, a researcher can apply on different kinds of soil from the different countries. This may able to increase the crop yield around the world. So for the last question is the method applicable for the farmers from the rural areas to apply the PGPR in their plantation without any supports of technologies? In the studies, 10 different types of soil bacteria strains isolated from the wheat reservoir were used. Out of the 10 bacteria strains, 5 strains were identified from the genus Pseudomonas, 4 strains belongs to the genus Bacillus, and 1 strain belongs to the genus Alcalgenesis. So as for suggestion, the local government is encouraged to support the local farmers from rural areas by providing them a number of PGPR for them to and teach them to culture the bacteria, etc. So that's all from me. Thank you.
And with that, we have reached to the conclusion and the end of this presentation. Thank you so much for your time.